Hello there, my name is Benito and I'm here to do a revision video for the AQA syllabus of AS Biology. Now, I'm nowhere near an expert because I'm actually studying my AS Biology at the moment, but hopefully this will help you because it's going to help me to revise. So that's the plan. Let's see what happens in this chaos. So, what we're doing, through popular demand, we're going to do Chapter 3, which is called Cells and Movement in and Out of Them. So what we're going to do, we're going to look at the structure of a cell and how substances are transported in and out of them. Yeah. But first we're going to have a look at how we can actually see cells. That's my hand, by the way. How we can actually see cells, and obviously that's going to be through a microscope. Now, different microscopes have different magnifications, and magnification actually has its own little formula. Magnification is the size of the image over the object. Size of the image over size of the object, yeah. So for example, let's say this is an image, right? This is a photograph of a cell. And I told you the length from end to end was, oh god, what would you say that was? 15 centimeters. Yeah. But its actual length was, I don't know, this probably isn't accurate, but 10 micrometers. Now, the biggest place to slip up on here is getting your units wrong, because obviously we've got centimeters here and micrometers down here. We have to convert them into the same units. Now personally, just for convenience, I would convert them both into meters, because that's in all SI units. But as it's a division, it's a ratio, it doesn't really matter just as long as they're the same units. But what we've got to know is how much micro is in relation to a meter. Well, we know that micrometer is the same, 1 micrometer is the same as 1 times 10 to the minus 6 meters. So, to get that into meters, we're going to do 10 times 10 to the minus 6. Centimeters, we should know how to convert into meters. We're just going to divide by 100. Yeah, that's right, isn't it? I'm not being stupid. No, I'm not. Okay. So, I'm not going to do the full calculation because I can't be bothered, but the size of the image is 15 over 100. Over 10 times 10 to the minus 6, and that's your magnification. And that makes sense because it's obviously going to be image divided by object. We're seeing how much the image has got enlarged, the object has got enlarged. So it's a ratio. If it was object over image, well, obviously the object is the smaller number. So you're going to get a very small magnification, less than 1. So that's not going to make a bit of sense whatsoever. So it's obviously going to be image over object. Another way you can remember it is that I comes before O in the alphabet. There you go, job done. Now we can also look at different types of microscope. Now there's the electron microscope. Now electrons are very useful because they have a very small wavelength. It's actually called the de Broglie wavelength, but that's physics. Yeah, I do physics as well. So as electrons have a very small wavelength, that means they have a very good resolving power. Resolution is very hard to define, but it's sort of how well you can distinguish between two objects in an image. I mean, if two really small things are close together, it's going to be really close. It's going to be really difficult to distinguish whether they're actually two objects or just one solid thing, if you haven't got a very good microscope. So that's why electron microscopes are useful. Now there's two types. There's the scanning electron microscope, and then there's the transmission electron microscope, the TEM and the SEM. The transmission electron microscope has a much higher resolving power, so that's an advantage for that. This is basically all they're going to ask you really in this, but the disadvantage is that the the samples need to be extremely thin for that. So yeah. And the scanning electron microscope, they don't need to be as thin, so that's an advantage for the scanning electron microscope. Obviously, it has a lower resolving power. And of course, for a transmission electron microscope, it all has to be in a vacuum, because otherwise the electrons would interact with molecules in the air, which wouldn't be very good. That's it in basic form. Now let's see the structure of a cell. Now, cells have different parts of them. The bits you have to know are the nucleus, and that contains basically what we're saying here is DNA, all the genetic information. The nucleus contains pores, which allows messenger RNA to be transported to ribosomes, so proteins can be produced using the coded information from the DNA. Pardon me. Mitochondria. They're basically the main sites of respiration. Now, respiration is what gives us energy. Basically, it produces energy in the form of ATP, which basically keeps us moving. So it converts glucose and oxygen into carbon dioxide and water, plus ATP. Very useful. It's sort of made up. It's sort of disc-shaped, but it's got sort of indents in it. They're called cristae, and the fluid inside is called the matrix. And that is providing a huge surface area for enzymes to attach to catalyze the reaction even further to produce even more energy, which is really good. The endoplasmic reticulum, there's two types of endoplasmic reticulum. There's the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum is rough because it contains lysosomes. No, it contains ribosomes, which are used, as I said, in protein synthesis. The smooth the smooth endoplasmic reticulum sort of synthesizes and stores and transports lipids and carbohydrates. That's basically all you have to know about them. You also have the Golgi apparatus. I can't spell. Right. And that basically forms the lysosomes. And it adds certain bits of proteins to make glycoproteins, which can be used in the cell surface membrane, and so on and so on and so on. And they form lysosomes by forming little vesicles. Bits break off the Golgi apparatus. And the lysosomes, you're probably wondering what they're all about. They contain enzymes, which basically break down. They're basically the bin men of the cell, really, because they break down all the stuff. They're used in phagocytosis, which is used as non-specific immunity. So they're very useful. So they release lots of enzymes, kill lots of things we don't want. Hooray. We've also obviously got ribosomes, which we've already mentioned. They're the sites of protein synthesis. There's two types, 70S type and 80s type, as you can probably suggest. 80s type is bigger. They're found in eukaryotic cells. Yeah, that's something else I should have mentioned. That's the eukaryotic cells. Us, basically. Us, plants, I think fungi are eukaryotic. I'm not sure. Anyway, basically bacteria and stuff, they'll have 70s type. Because they're smaller. They don't have as much room, as it were. Microvilli, on an epithelial cell, you'll have microvilli. These are projections on the cell. If I was to draw my cell here, you see all the bumps. And the basic reason to this is surface area. Because if you have loads of microvilli, and you're probably on these, they're probably villi, you'll have loads of little projections on here as well. Because if you're an epithelial cell in the small intestine, you have got a lot of area there to absorb all those substances which will eventually go into the bloodstream and will then be broken down into substances like glucose, which can be used in respiration, blah, 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 blah. And that's basically it. So you can tell what the main function of a cell by looking how much of specific organelles it has. For example, if it has lots of mitochondria, you could predict that it has a high metabolic activity, so it could be a muscle cell or something like that. If it has loads of microvilli, you could predict that it was a cell in the small intestine for absorbing stuff. Well, that's basically all we've got time for. Yeah, that's a bit of a shame. I hope that was useful. I'll do another one in a minute. But, see ya.